Okay, so as I said, we did this benchmark. Yeah, it's already a long time ago, and back then we found these classifiers, right? They were all designed to classify single cell data. Some of them you will experiment with today, if I'm not mistaken. So SC-Predict, Single R, and the one from CEURAT. So maybe a word on that. So Single R is sort of a lazy classifier. So what they do is that they take bulk RNA-seq data, mostly, that are of purified cell types. And then for each cell that you have in your single cell data, they calculate the correlation between the expression profile of your cell and the expression profile of each pure cell type from the bulk data. So it is a sort of a nearest neighbor classification, right? You try to look for the correlation, which is a measure of distance between the cell that you're interested in and cells from the representatives from the reference, right? And then you match it to the one that is mostly correlated. There are a few steps in between on, filtering correlations and so on, but it is simply looking at correlation between the expression profile of your cell and the expression profile of reference data. SC-Predict is a support vector machine. They have a smart way to select the features they use for classification, but it is simply a support vector machine classifier, as we explained now. CEURAT is again using a lazy classifier. So there's actually no classification. There's no training of the data. So you have your reference map, you do integration. I believe you covered the integration already in the course. Yeah. So you do the integration, then you have the query and the reference in the same place. And then they use a weighted classifier like we explained here, like this one. So you basically look at the distances to for each query cell, you look at the distances to the reference cells and you weight them by the distance. So the ones close to you get a high weight, the ones far away, you get a low weight. And then I believe they do a majority vote or something like that. Yeah. Is this clear so far? Okay, so what we did for the benchmark, we actually grouped these classifiers into groups like Moana and SC-Predict. They are both SVM classifiers with some variations. SC-Map has two versions. One of them is SC-Map Cell, that's a KNN, so it looks at K nearest neighbors. SC-Map Cluster is a nearest mean classifier. These are all random forest. These are neural network based. This is an LDA and so on. And these are ones that we cannot actually classify into any classical classification scheme. What we also did is that we took these basic classifiers. So as I said, like for instance, these are based on SVM. So we can also just take the SVM from scikit-learn in Python and then use it, right? There's nothing against this. The only difference is that these tweaked classifiers have very specific choices. Why did we do that? If you work on developing a new classifier, and we certainly do this mistake, so I'm not criticizing anyone here, you really try to make it perfect, right? So let's assume you're working on 5 data sets, so you keep tweaking your classifier so it's perfect for these 5 data sets. But if I now take this classifier and apply it to another 30 data sets, it will probably not work, because the tweaking you have done is really specific to these 5 data sets. No matter how large the group of data sets that you look at, you will be making choices that do not generalize well. And that's why we thought, if you throw in these off-the-shelf classifiers, they have never been tweaked for single cell data, they might actually do well, because they will generalize much better than the specific classifiers.